Hello there, and welcome to the Deep Somna. Tonight, we're embarking on a journey to explore one of nature's most fascinating creations, the human body. It holds far more secrets than we could ever touch upon in this journey. If your eyes begin to grow heavy, that's perfectly natural. Listen for as long as you'd like, or use this as background music as you drift off to sleep. I hope that by the end of this narrative, you'll have learned something new. Now, please join me as we marvel together at the miraculous nature of the human body. Let's begin. Somewhere beneath the surface of awareness, a universe unfolds in silence. Not the universe of stars and void, but something closer. Something woven into the fabric of bone and breath. The kind of universe that exists in the space between one heartbeat and the next. In the dark interior where blood moves without instruction. Where cells divide without permission. Where the body maintains its own deep rhythms independent of thought or will. Right now in this moment, approximately 2 million red blood cells have just died. 2 million more have been born to replace them. This exchange happened without fanfare, without sensation. The body's bone marrow, hidden deep within the hollows of the pelvis and ribs, is manufacturing these cells at a rate that defies easy comprehension. Each second, nearly 3 million new red blood cells emerge from their birthplace, fully formed and ready to carry oxygen through the winding channels of the circulatory system. They will live for about 4 months, traveling through the body countless times before they too are quietly dismantled and their components recycled into new forms. The marrow itself is a peculiar substance, soft and fatty, unlike the hard mineral exterior of bone that houses it. Within this protected interior space, stem cells undergo a transformation, differentiating into the various types of blood cells the body requires. Some become red blood cells, others white blood cells tasked with immunity. Still others become platelets that help blood clot when vessels are damaged. The process is called hematopoiesis, a word that sounds ancient and ceremonial, which perhaps it should, given that this has been occurring in vertebrate bodies for hundreds of millions of years, long before humans walked upright, long before there were forests or flowers or even fish with jaws. Blood itself is stranger than it appears. It is not a simple fluid but a tissue, a living tissue that flows. Within every drop are millions of cells suspended in plasma, that yellowish liquid that remains when all the cellular components are removed. Plasma is mostly water, but it carries within it a complex mixture of proteins, hormones, nutrients, waste products, and dissolved gases. It is the body's primary transport medium, the river upon which all other biological processes depend. Without it, cells would starve in their own neighborhoods, unable to receive glucose or oxygen, unable to export the carbon dioxide and metabolic waste they produce. The heart that moves this blood is itself a marvel of perpetual motion. Unlike skeletal muscles that can rest, that can be still, the cardiac muscle has been contracting since before birth, since the embryonic heart began to beat at around 3 weeks of development, when the embryo itself was barely visible to the naked eye. That first beat occurred in a body no larger than a grain of rice, and it has not stopped since. Will not stop until the very end. The heart does not fatigue in the way other muscles do. It has its own internal pacemaker, a cluster of specialized cells in the right atrium that generate electrical impulses approximately 70 times per minute, though this rate varies with activity, emotion, and sleep. Each contraction follows a precise sequence. the atria, the upper chambers, fill with blood and squeeze, pushing their contents down into the ventricles below. Then the ventricles contract with greater force, sending blood out through the major arteries. The right ventricle pushes blood toward the lungs, where it will release carbon dioxide and pick up fresh oxygen. The left ventricle, thicker and more muscular, propels blood out into the aorta and from there into the vast network of arteries that branch and branch again, becoming smaller and smaller until they transition into capillaries so narrow that red blood cells must pass through in single file, their flexible membranes deforming to squeeze through passages barely wider than the cells themselves. It is in these capillaries, these microscopic vessels with walls only 1 cell thick, that the real work happens. Oxygen diffuses out of the red blood cells, crosses the thin capillary wall, and enters the surrounding tissue. Carbon dioxide, the waste product of cellular respiration, makes the reverse journey, entering the blood to be carried back to the lungs and exhaled. This exchange is not active or forced. It happens through diffusion, molecules moving from areas of high concentration to areas of low concentration, following gradients established by the constant consumption of oxygen and production of carbon dioxide in living cells. The lungs themselves are an architecture of branching. The trachea divides into 2 bronchi, 1 for each lung. Each bronchus divides again and again, becoming smaller bronchioles, which eventually terminate in clusters of tiny air sacs called alveoli. There are approximately 500 million alveoli in the average adult lung, creating a surface area that, if spread flat, would cover roughly the size of a tennis court. This enormous surface area is necessary because gas exchange, even though it happens quickly at the molecular level, requires vast expanses of thin membrane for the body's needs to be met. Every breath brings fresh air into contact with this delicate surface, and every exhalation removes the stale air laden with carbon dioxide. Breathing happens mostly without thought. The diaphragm, a dome-shaped muscle beneath the lungs, contracts and flattens, expanding the chest cavity and drawing air inward. When it relaxes, the chest cavity shrinks and air is pushed out. This rhythm is controlled by the brainstem, specifically an area called the medulla oblongata, which monitors carbon dioxide levels in the blood and adjusts breathing rate accordingly. It is possible to override this automatic control, to hold the breath or breathe faster than necessary, but only for a time. Eventually, the brainstem reasserts control, and breathing resumes its natural pattern. The air that enters the lungs is not pure oxygen. Earth's atmosphere is approximately 78% nitrogen, 21% oxygen, and small amounts of other gases including argon and carbon dioxide. The nitrogen is largely inert, passing in and out of the lungs without being absorbed or used. It is the oxygen that matters, the oxygen that binds to hemoglobin molecules inside red blood cells. 4 oxygen molecules per hemoglobin, turning the blood a bright scarlet color. The oxygenated blood is darker, more maroon than red, though the common belief that it is blue is a misconception arising from the way veins appear through the skin. Hemoglobin is a protein, a large folded molecule, with a core structure built around iron atoms. It is the iron that actually binds the oxygen. It is the iron that gives blood its color. When oxygen binds, the iron shifts slightly in its molecular position, altering the light-absorbing properties of the hemoglobin and producing that characteristic red. Each red blood cell contains approximately 270 million hemoglobin molecules, and each hemoglobin can carry 4 oxygen molecules, which means a single red blood cell can transport over 1 billion oxygen molecules at once. Multiply this by the 25 trillion red blood cells in the human body, and the total oxygen carrying capacity becomes almost incomprehensible. Iron itself is an element forged in stars, not in the ordinary fusion that powers stars like the sun, which fuses hydrogen into helium and helium into heavier elements like carbon and oxygen, but in the final moments of massive stars, those giants many times larger than the sun, which in their death throws fuse elements all the way up to iron before collapsing and exploding in supernovae. The iron in hemoglobin, the iron that makes blood red, was created billions of years ago in stellar explosions, scattered into space, eventually coalescing into the disk of dust and gas that became the solar system, incorporating into the rocks of early Earth, dissolving into ancient oceans, taken up by simple organisms, and passed down through evolutionary lineages until it ended up here, circulating through veins and arteries, binding oxygen, sustaining life. This cosmic inheritance extends beyond iron. Carbon, the backbone of all organic molecules, was also forged in stars. Calcium in bones, phosphorus in DNA, sulfur in proteins, all these elements were created in stellar interiors and released into space when those stars died. The human body is made of star matter in the most literal sense, atoms that once existed in the hearts of suns, now arranged into the intricate structures of cells and tissues. Cells themselves are difficult to truly comprehend. They are the fundamental units of life, the smallest structures that can be said to be alive. A typical human cell is about 10 micrometers across, small enough that 100 of them laid side by side would barely span the width of a human hair. Yet within that microscopic volume exists a staggering complexity: the cell membrane, a double layer of lipid molecules studded with proteins, controls what enters and exits. Inside, the cytoplasm holds organelles, each with specialized functions. Mitochondria generate energy. Ribosomes assemble proteins. The endoplasmic reticulum and Golgi apparatus process and package molecules for transport. And at the center, the nucleus contains the genetic material. DNA coiled into chromosomes. DNA is often described as a blueprint, but that metaphor does not quite capture what it is. It is more like a library of instructions, a chemical archive written in a 4-letter alphabet composed of nucleotide bases: adenine, thymine, guanine, and cytosine, abbreviated A, T, G, and C. These bases pair in specific ways, A with T and G with C, forming the rungs of the famous double helix structure. The sequence of these bases along the DNA strand encodes genes, segments that contain the information needed to build proteins. Proteins are the workhorses of the cell, performing nearly every function necessary for life, from catalyzing chemical reactions to providing structural support to transporting molecules across membranes. Each human cell contains approximately 2 meters of DNA, tightly coiled and packaged into the nucleus. If all the DNA in all the cells of the human body were laid end to end, it would stretch from Earth to the Sun and back several hundred times. Yet this vast molecular thread is not static. It is constantly being accessed, read, copied, and repaired. Enzymes move along the DNA strand, unzipping the double helix to expose the genetic code. Other enzymes transcribe this code into RNA, a single-stranded molecule that carries the genetic information out of the nucleus and into the cytoplasm, where ribosomes use it as a template to assemble amino acids into proteins. The process is called the central dogma of molecular biology, DNA to RNA to protein. It is the fundamental mechanism by which genetic information is expressed, how the instructions encoded in the genome are translated into the physical structures and functions of the body. But it is not a simple one-way flow. Cells regulate which genes are active and which are silenced, responding to signals from their environment, from neighboring cells, from hormones circulating in the blood. A liver cell and a neuron contain the same DNA, the same genetic information, yet they are utterly different in form and function because they express different subsets of genes. Neurons are among the most remarkable cells in the body. They are electrically active, capable of generating and transmitting signals over long distances. A neuron consists of a cell body containing the nucleus, dendrites that receive signals from other neurons, and an axon that transmits signals onward. Some axons are extremely long. The neurons that control muscles in the feet have axons that stretch all the way from the lower spinal cord down the length of the leg, a distance that can exceed a meter. These axons are wrapped in a fatty insulating sheath called myelin, which allows electrical signals to travel faster by enabling a kind of leapfrog transmission called saltatory conduction, where the signal jumps from gap to gap in the myelin sheath rather than propagating continuously along the axon. The electrical signals themselves, called action potentials, are generated by the movement of ions across the neuron's membrane. At rest, the inside of a neuron is negatively charged relative to the outside, maintained by pumps that actively transport sodium ions out and potassium ions in. When the neuron is stimulated, channels in the membrane open, allowing sodium to rush inward, briefly reversing the charge. This reversal propagates along the axon like a wave, reaching the axon terminal, where it triggers the release of neurotransmitters, chemical messengers that cross the narrow gap, the synapse, between neurons and bind to receptors on the next cell, either exciting it or inhibiting it, depending on the type of neurotransmitter and receptor involved. The brain contains roughly 86 billion neurons, each forming thousands of synaptic connections with other neurons, creating a network of unimaginable complexity. The total number of synapses is estimated to be around 100 trillion, a number so large it exceeds the number of stars in the Milky Way galaxy. These connections are not fixed. They change constantly, strengthening with repeated use, weakening when neglected, forming entirely new connections when new information is learned. This plasticity is the basis of memory and learning, the physical substrate of thought and experience. Memory itself is not stored in a single location, but distributed across networks of neurons. When a memory is formed, a pattern of synaptic connections is established. Recalling that memory reactivates the same pattern, though not perfectly. Each time a memory is recalled, it is subtly altered, reconsolidated with slight modifications. This is why memory is fallible, why eyewitness testimony can be unreliable, why the past as we remember it is not an exact recording but a reconstruction. A story the brain tells itself based on fragments of encoded experience. Sleep plays a crucial role in this process. During sleep, particularly during the stage known as slow wave sleep, The brain replays patterns of activity from waking hours, consolidating memories, integrating new information with existing knowledge. The hippocampus, a structure deep within the brain essential for forming new memories, communicates with the cortex, the brain's outer layer where long-term memories are ultimately stored. Dreams, which occur mostly during REM sleep, a stage characterized by rapid eye movements and heightened brain activity, may serve a different function, processing emotions, simulating scenarios, perhaps even solving problems by making novel connections between disparate pieces of information. The brain never fully rests. Even in the deepest stages of sleep, neural activity continues. The brainstem maintains vital functions like breathing and heart rate. The thalamus, a relay station for sensory information, remains active, though it filters out most external stimuli to prevent waking. The default mode network, a set of brain regions active when not focused on external tasks, engages in self-referential thought, mind-wandering, internal simulation. The sleeping brain is not unconscious in the sense of being inactive. It is differently conscious, turned inward, processing and organizing, preparing for wakefulness. The transition between wakefulness and sleep is mediated by complex interactions between neurotransmitters and hormones. Adenosine, a byproduct of cellular energy use, accumulates in the brain during waking hours, gradually increasing sleep pressure. Caffeine works by blocking adenosine receptors, temporarily preventing the sensation of sleepiness. Melatonin, a hormone produced by the pineal gland in response to darkness, promotes sleep by acting on receptors in the brain. The circadian rhythm, the body's internal 24-hour clock, coordinates sleep-wake cycles with the external environment, primarily through light exposure detected by specialized cells in the retina that signal the suprachiasmatic nucleus, the brain's master clock. Light itself is a stream of photons, particles of electromagnetic radiation that travel at 300 million meters per second. When photons enter the eye, they pass through the cornea and lens, which focus them onto the retina, a layer of light-sensitive tissue at the back of the eye. The retina contains two types of photoreceptor cells: rods, which are sensitive to dim light and provide black and white vision, and cones, which detect color and function best in bright light. There are three types of cones, each sensitive to different wavelengths of light, corresponding roughly to red, green, and blue. The brain combines signals from these cones to perceive the full spectrum of colors. The retina does more than detect light. It performs significant processing before sending information to the brain. Photoreceptors connect to bipolar cells, which connect to ganglion cells, whose axons form the optic nerve. But there are also horizontal cells and amacrine cells that integrate signals laterally, enhancing contrast and detecting edges. Some ganglion cells are specifically tuned to detect motion, others to detect specific orientations of lines. This means that even before visual information reaches the visual cortex, it has already been analyzed and refined, patterns extracted, features highlighted. The visual cortex, located at the back of the brain, continues this processing. Different regions specialize in different aspects of vision. One area detects edges and orientations. Another processes color. Yet another recognizes motion. There are even regions specialized for recognizing faces, a crucial social function that humans perform with remarkable speed and accuracy. Damage to these areas can result in prosopagnosia, an inability to recognize faces, even familiar ones, even one's own reflection, though other aspects of vision remain intact. Vision feels immediate and effortless, but it is the result of extensive computation. The brain does not passively receive images like a camera. It actively constructs a visual experience from incomplete and often ambiguous information. It fills in the blind spot where the optic nerve exits the retina. It stabilizes the visual world despite constant eye movements. It infers three-dimensional structure from two-dimensional retinal images. It compensates for varying lighting conditions to maintain color constancy. The reality we see is not the world as it is, but a model the brain builds, a best guess based on sensory input and prior experience. This applies to all senses. Hearing, for instance, begins with sound waves, vibrations in the air that enter the ear canal and strike the eardrum, a thin membrane that vibrates in response. These vibrations are transmitted through three tiny bones in the middle ear, the malleus, incus, and stapes, the smallest bones in the human body, which amplify the vibrations and transfer them to the cochlea, a spiral-shaped structure in the inner ear, filled with fluid. Inside the cochlea is the organ of Corti, lined with thousands of hair cells, each tuned to a specific frequency. When the fluid in the cochlea vibrates, it causes these hair cells to bend, triggering electrical signals that travel along the auditory nerve to the brain. The brain interprets these signals as sound, but it does more than that. It identifies the location of sounds by comparing the timing and intensity of signals reaching each ear. It filters out background noise to focus on relevant sounds, a phenomenon known as the cocktail party effect. It recognizes patterns, distinguishing speech from music from environmental noise. It associates sounds with meanings, an arbitrary mapping learned through experience, which is why language is possible, why a sequence of sounds can represent objects, actions, abstract concepts. Touch is perhaps the most distributed sense. The entire surface of the skin, the body's largest organ, is equipped with sensory receptors. Some detect light touch, others pressure, still others vibration, temperature, or pain. These receptors are not evenly distributed. The fingertips, lips, and tongue have extremely high densities of touch receptors, making them exquisitely sensitive, while the back and legs have far fewer. This uneven distribution is reflected in the brain's sensory map, where disproportionately large areas are devoted to processing signals from the hands and face. The skin itself is a complex organ, composed of multiple layers. The outermost layer, the epidermis, is made mostly of dead cells filled with keratin, a tough protein that provides protection. These cells are constantly shed and replaced from below. Beneath the epidermis lies the dermis, a thicker layer containing blood vessels, nerve endings, hair follicles, and sweat glands. The dermis provides structural support and elasticity, thanks to collagen and elastin fibers. Below that is the hypodermis, a layer of fat that insulates, cushions, and stores energy. Sweat glands play a vital role in temperature regulation. The human body generates heat as a byproduct of metabolism, and maintaining a stable internal temperature is essential for proper enzyme function and cellular processes. When body temperature rises, whether from exercise, fever, or external heat, the hypothalamus, the brain's temperature control center, triggers sweating. Sweat, primarily water with dissolved salts, is secreted onto the skin surface, where it evaporates, removing heat and cooling the body. Humans are among the best endurance athletes in the animal kingdom, in part because of this highly effective cooling system, which allows sustained physical activity even in hot environments. Temperature regulation also involves blood flow to the skin. When too warm, blood vessels near the surface dilate, bringing more blood close to the skin where heat can radiate away. When too cold, these vessels constrict, reducing blood flow to the surface and conserving heat for the core. Shivering generates additional heat through rapid involuntary muscle contractions. Goosebumps, a vestigial response from ancestors with more body hair, occur when tiny muscles at the base of hair follicles contract, attempting to fluff up insulation that no longer exists. Hair itself is made of keratin, the same protein that forms the outer layer of skin and nails. Each hair grows from a follicle, a small pit in the skin's surface. At the base of the follicle, cells divide rapidly, pushing older cells upward. As these cells move away from their blood supply, they die and harden, forming the hair shaft. Hair grows in cycles, a growth phase lasting several years, a brief transitional phase, and a resting phase before the hair falls out and the scalp grows continuously for years, which is why it can become very long, while body hair has shorter growth phases and remains relatively short. Nails grow from a similar process, cells dividing in the nail matrix beneath the cuticle, hardening as they push forward. Fingernails grow faster than toenails, approximately 3 millimeters per month, and nails on the dominant hand tend to grow faster than those on the non-dominant hand, possibly due to increased blood flow from more frequent use. Nails protect the sensitive tips of fingers and toes and provide support for fine manipulation tasks. Bones, too, are living tissue, constantly being remodeled. Cells called osteoclasts break down old bone, while osteoblasts build new bone in its place. This turnover allows bones to adapt to mechanical stress, becoming denser and stronger where needed, shedding mass where it is not. It also allows bones to heal after fractures. When a bone breaks, blood vessels are damaged, and a hematoma forms at the site. Inflammatory cells arrive to clean up debris. Then, a soft callus of cartilage forms, bridging the gap. Finally, osteoblasts move in and replace the cartilage with new bone, which is gradually remodeled until the fracture site is nearly indistinguishable from the surrounding bone. Bones serve multiple functions beyond structural support. They protect vital organs. The skull encases the brain. The rib cage shields the heart and lungs. The vertebrae surround the spinal cord. They act as levers for muscles, enabling movement. They store minerals, particularly calcium and phosphorus, releasing them into the bloodstream as needed to maintain proper levels for nerve and muscle function. And as mentioned earlier, they house bone marrow, the site of blood cell production. Muscles, the engines of movement, are composed of bundles of muscle fibers, specialized cells that contract when stimulated by motor neurons. Each muscle fiber contains myofibrils, long strands made of repeating units called sarcomeres. Within each sarcomere are two types of protein filaments. thick filaments made of myosin and thin filaments made of actin. When a nerve signal reaches the muscle, calcium ions are released inside the muscle fiber, allowing myosin heads to attach to actin filaments and pull, shortening the sarcomere. When many sarcomeres shorten simultaneously, the entire muscle contracts. This process requires energy in the form of ATP, adenosine triphosphate, the universal energy currency of cells. ATP is produced primarily in the mitochondria through a process called cellular respiration, which uses oxygen and glucose to generate ATP, releasing carbon dioxide and water as byproducts. Mitochondria are often called the powerhouses of the cell, and a single muscle cell can contain thousands of them, providing the enormous amounts of energy needed for sustained contraction. Mitochondria have their own DNA, separate from the nuclear DNA, and they reproduce independently within cells. This is because mitochondria are thought to have originated as free-living bacteria that were engulfed by early eukaryotic cells in a symbiotic relationship that became permanent over evolutionary time. The mitochondrial DNA is inherited exclusively from the mother, passed down through the egg cell, providing a direct maternal lineage that can be traced back through generations. This endosymbiotic origin extends to chloroplasts in plant cells, which perform photosynthesis, converting sunlight into chemical energy. Though human cells lack chloroplasts, the oxygen we breathe and the food we eat ultimately depend on photosynthesis. Plants take in carbon dioxide and water, using sunlight to convert them into glucose and oxygen. We consume the glucose either directly by eating plants or indirectly by eating animals that ate plants, and we breathe the oxygen, completing a cycle that links all aerobic life on Earth. The digestive system is where food is broken down into molecules small enough to be absorbed into the bloodstream. Digestion begins in the mouth, where teeth mechanically break food into smaller pieces in saliva. Secreted by salivary glands, begins the chemical breakdown of starches using an enzyme called amylase. The tongue, a muscular organ covered in taste buds, manipulates the food and helps form it into a bolus, a soft mass that can be swallowed. Swallowing is a complex, coordinated action involving multiple muscles and precise timing. The tongue pushes the bolus backward into the pharynx, triggering a reflex that closes the airway and opens the esophagus. The bolus is then propelled down the esophagus by peristalsis, rhythmic muscular contractions that push food toward the stomach. At the end of the esophagus, a sphincter relaxes to allow the bolus to enter the stomach, then closes to prevent stomach acid from flowing back up. The stomach is a muscular sac that churns food, mixing it with gastric juices secreted by cells in the stomach lining. These juices include hydrochloric acid, which creates an acidic environment harsh enough to kill most bacteria and begin breaking down proteins, and pepsin, an enzyme that digests proteins into smaller peptides. The stomach lining is protected from this acid by a thick layer of mucus, which is constantly renewed. Despite this protection, the cells lining the stomach are replaced every few days, one of the fastest turnover rates in the body. After several hours, the partially digested food, now a thick liquid called chyme, is released in small amounts into the small intestine, where most nutrient absorption occurs. The small intestine is approximately 6 meters long, though its name comes from its narrow diameter compared to the large intestine. Its inner surface is covered with millions of tiny finger-like projections called villi, which increase the surface area available for absorption. Each villus is covered with even smaller projections called microvilli, creating a surface area that, if flattened, would cover roughly the size of a tennis court. Enzymes from the pancreas and bile from the liver continue the digestive process in the small intestine. The pancreas secretes enzymes that break down carbohydrates, proteins, and fats. Bile, produced by the liver and stored in the gallbladder, emulsifies fats, breaking them into smaller droplets that enzymes can act upon more easily. As nutrients are broken down into simple sugars, amino acids, and fatty acids, they are absorbed through the walls of the small intestine into the bloodstream and lymphatic system, eventually reaching cells throughout the body. The liver, located in the upper right abdomen, is one of the largest and most metabolically active organs. It receives nutrient-rich blood directly from the small intestine via the hepatic portal vein. Here, the liver processes these nutrients, storing excess glucose as glycogen, converting amino acids, detoxifying harmful substances, and producing bile. The liver performs over 500 distinct functions, making it indispensable. Unlike most organs, the liver has a remarkable capacity for regeneration. If up to 75% of the liver is removed or damaged, it can regrow to its original size within months. What remains of the chyme after nutrient absorption passes into the large intestine, also called the colon, which is wider but shorter than the small intestine, approximately 1.5 meters long. The colon's primary function is to absorb water and electrolytes from the remaining indigestible material, consolidating it into feces. The colon is also home to trillions of bacteria, the gut microbiome, which ferment indigestible fibers, producing vitamins like vitamin K and certain B vitamins, and generating short-chain fatty acids that nourish the cells lining the colon. This microbial community is vast and diverse, containing hundreds of different species of bacteria, along with smaller populations of archaea, fungi, and viruses. The composition of this microbiome varies from person to person and is influenced by diet, environment, genetics, and even birth method. Babies born vaginally are colonized by their mother's vaginal and fecal bacteria, while those born via cesarean section acquire different initial communities. This early colonization shapes immune development and may influence long-term health. The relationship between host and microbiome is symbiotic. The bacteria gain a stable environment and steady food supply. The host gains assistance with digestion, synthesis of essential nutrients, protection against pathogens, and modulation of the immune system. Some gut bacteria communicate with the nervous system via the vagus nerve, influencing mood and behavior, which is why the gut is sometimes called the second brain. The enteric nervous system, embedded in the walls of the digestive tract, contains over 100 million neurons, more than the spinal cord, and can function independently of the central nervous system, controlling digestion through local reflexes. Waste products that are not excreted through the digestive system are filtered by the kidneys. The kidneys are bean-shaped organs located on either side of the spine, just below the rib cage. Each kidney contains approximately 1 million nephrons, microscopic filtering units that remove waste from the blood while retaining essential substances. Blood enters the nephron through a cluster of capillaries called the glomerulus, where pressure forces water and small molecules out of the blood and into a tubule. As this filtrate moves through the tubule, useful substances like glucose, amino acids, and most of the water are reabsorbed back into the blood. What remains, now called urine, contains urea, excess salts, and other wastes. Urine flows from the kidneys through ureters into the bladder, a muscular sac that stores urine until it is convenient to release it. The bladder can hold several hundred milliliters of urine, stretching as it fills. When full, stretch receptors in the bladder wall send signals to the brain, creating the sensation of needing to urinate. Voluntary control, learned in childhood, allows the external sphincter to remain closed until an appropriate time and place for urination. The kidneys also play crucial roles in regulating blood pressure, maintaining electrolyte balance, and stimulating red blood cell production. When oxygen levels in the blood are low, the kidneys release a hormone called erythropoietin, which signals the bone marrow to increase red blood cell production. They also regulate the balance of sodium, potassium, calcium, and other ions, which is essential for nerve and muscle function, and they control blood volume and pressure by adjusting how much water is reabsorbed or excreted. Hormones, chemical messengers produced by endocrine glands, coordinate functions throughout the body. The pituitary gland, a pea-sized structure at the base of the brain, is often called the master gland because it secretes hormones that regulate other endocrine glands. Growth hormone stimulates growth and cell reproduction. Thyroid-stimulating hormone prompts the thyroid gland to release hormones that regulate metabolism. Adrenocorticotropic hormone triggers the adrenal glands to produce cortisol, a stress hormone that affects metabolism, immune function, and blood pressure. The thyroid gland, located in the neck, wraps around the trachea and produces thyroid hormones that regulate the rate at which cells convert nutrients into energy. Too much thyroid hormone causes metabolism to speed up, leading to weight loss, rapid heartbeat, and nervousness. Too little slows metabolism, causing fatigue, weight gain, and sensitivity to cold. The thyroid also produces calcitonin, which helps regulate calcium levels in the blood by inhibiting bone breakdown. The adrenal glands, small triangular structures sitting atop each kidney, produce several important hormones. The outer layer, the adrenal cortex, produces cortisol, aldosterone, which regulates sodium and potassium balance, and small amounts of sex hormones. The inner layer, the adrenal medulla, produces adrenaline and noradrenaline, hormones that prepare the body for fight or flight responses by increasing heart rate, blood pressure, and energy availability. The pancreas functions both as an exocrine gland, secreting digestive enzymes into the small intestine, and as an endocrine gland, releasing hormones into the bloodstream. Clusters of cells called islets of Langerhans produce insulin and glucagon, hormones that regulate blood glucose levels. Insulin lowers blood glucose by promoting its uptake into cells and storage as glycogen. Glucagon raises blood glucose by stimulating the breakdown of glycogen in the liver. This balance is critical. Too much glucose damages blood vessels and nerves, while too little deprives cells of energy, particularly brain cells, which rely almost exclusively on glucose. The reproductive system is controlled by a complex interplay of hormones. In males, the testes produce testosterone, which drives the development of male characteristics and the production of sperm. Sperm cells are among the smallest cells in the body, consisting of a head containing genetic material, a midpiece packed with mitochondria for energy, and a tail that propels the cell forward. Sperm production, or spermatogenesis, begins at puberty and continues throughout life, though it declines with age. In females, the ovaries produce eggs and secrete estrogen and progesterone. Unlike sperm, which are produced continuously, all the eggs a woman will ever have are present at birth, though in an immature form. Starting at puberty, typically one egg matures each month during the menstrual cycle. Hormones from the pituitary gland trigger the development of a follicle in the ovary, which releases the egg in a process called ovulation. If fertilized by a sperm, the egg can implant in the uterus and develop into an embryo. If not, hormone levels drop, triggering menstruation, the shedding of the uterine lining. Fertilization, when it occurs, usually happens in the fallopian tube. The sperm must travel through the cervix, into the uterus, and up the fallopian tube to reach the egg. Of the millions of sperm released during ejaculation, only a few hundred typically reach the egg, and only one will successfully penetrate its outer membrane. Once a sperm enters, the egg's membrane changes to prevent others from entering. The genetic material from sperm and egg combine, forming a single cell with a complete set of chromosomes, half from each parent. This cell, the zygote, begins dividing immediately, traveling down the fallopian tube toward the uterus, while developing into a ball of cells called a blastocyst. Upon reaching the uterus, the blastocyst implants in the uterine lining, now thickened and enriched with blood vessels in preparation. Implantation marks the beginning of pregnancy. Cells differentiate rapidly, some forming the embryo, others the placenta, an organ that connects the developing fetus to the mother's blood supply, allowing nutrients and oxygen to pass from mother to fetus and waste products to pass in the opposite direction. The placenta also produces hormones that maintain pregnancy. Over the following weeks and months, the embryo transforms from a cluster of cells into a recognizable form. By week 4, the heart begins to beat, a simple tube that will fold and divide into chambers. By week 8, all major organs have begun to form, though they are not yet functional. The embryo is now called a fetus. Limbs elongate, fingers and toes separate, facial features become distinct. The brain grows rapidly, forming billions of neurons that migrate to their designated positions, guided by chemical signals and physical scaffolds. Fetal development follows a precise choreography, encoded in the genome but responsive to the environment. The mother's nutrition, stress levels, exposure to toxins, all influence development. The fetus floats in amniotic fluid, a protective cushion that maintains constant temperature and allows freedom of movement. The fetus swallows this fluid, practicing the swallowing reflex, and urinates into it, the kidneys already functioning. The fluid is constantly replenished and filtered by the placenta. As pregnancy progresses, the fetus grows larger, and its movements become stronger, perceptible to the mother. By the 3rd trimester, the fetus can hear sounds from outside the womb, muffled but recognizable. The mother's heartbeat, her voice, external noises, all register in the developing auditory system, Studies suggest that newborns can recognize their mother's voice and even specific sounds or music they were exposed to repeatedly before birth. The lungs are among the last organs to mature. Throughout most of pregnancy, the fetus does not breathe air. Oxygen comes from the mother's blood via the placenta. The lungs are filled with fluid and collapsed. Only in the final weeks do the lungs produce sufficient surfactant, a substance that prevents the alveoli from collapsing when exposed to air. At birth, the first breath is a dramatic transition. The infant must clear fluid from the lungs, expand them with air, and begin exchanging gases independently. This first breath often comes with a cry, a reflexive response to the sudden flood of sensory input and the effort of breathing. Birth itself is triggered by a cascade of hormonal signals from both mother and fetus. Contractions of the uterine muscle, driven by oxytocin, push the baby through the birth canal, a process that can take hours or even days for a first birth. The baby's skull bones are not yet fused, allowing them to overlap slightly, compressing to fit through the narrow passage. Once born, the baby must adapt rapidly to a radically different environment. Temperature regulation, digestion, breathing, all must function independently now. The newborn brain is remarkably underdeveloped compared to other mammals. A foal can walk within hours of birth. A human infant will take months to even lift its head reliably. This extended period of helplessness reflects the trade-off of being born with a brain large enough to eventually support complex cognition, but immature enough to fit through the birth canal. Most brain development occurs after birth. Neurons continue to migrate. Synapses form at an explosive rate, and experiences begin to shape neural circuits in ways that will persist throughout life. Early experiences have profound effects on brain development. Infants who receive consistent care, who are held, spoken to, and responded to, develop more robust neural connections in areas related to emotion regulation and social cognition. Neglect or trauma during these critical periods can alter brain architecture in lasting ways. The brain is most plastic in infancy and early childhood, though plasticity continues to some degree throughout life. Learning a language is easiest in childhood when the auditory cortex is highly sensitive to phonetic distinctions. Adults can still learn languages, but typically with more effort and less native-like pronunciation. Language itself is one of the most distinctly human capabilities. While other animals communicate, human language is unique in its complexity, flexibility, and generativity. With a finite set of sounds and grammatical rules, humans can produce and understand an infinite number of sentences, expressing abstract concepts, hypothetical scenarios, and elaborate narratives. Language appears to be supported by specialized brain regions, particularly Broca's area, involved in speech production, and Wernicke's area, involved in language comprehension, both typically located in the left hemisphere. How language evolved remains one of the great mysteries. It requires not only cognitive abilities, but also precise motor control of the vocal apparatus. The larynx, tongue, lips, and jaw must coordinate with extraordinary precision to produce the rapid sequences of sounds that make up speech. The human vocal tract is uniquely shaped for this purpose, though it comes at a cost. The low position of the larynx increases the risk of choking, as food and air passages cross in the pharynx. Speech production involves the lungs providing airflow, the larynx housing the vocal cords that vibrate to produce sound, and the articulators, tongue, lips, teeth, and palate, shaping that sound into distinct phonemes. The tongue is one of the most flexible muscles in the body, capable of rapid and precise movements. The lips can form tight seals or small openings. The soft palate can rise to direct air through the mouth or lower to direct it through the nose, creating nasal sounds. All of this happens at remarkable speed. Fluent speakers can produce several syllables per second, each requiring dozens of coordinated muscle movements. The voice itself is produced by the vibration of the vocal cords, two folds of tissue stretched across the larynx. Air from the lungs passes between them, causing them to vibrate. The rate of vibration determines pitch. Tightening the cords increases pitch. Loosening them lowers it. The sound produced by the vocal cords is then modified by the resonance characteristics of the throat, mouth, and nasal cavities, which amplify certain frequencies and dampen others, creating the unique timbre of each person's voice. Everyone's voice is distinctive, shaped by the size and shape of their vocal tract, the length and tension of their vocal cords, and habitual patterns of articulation. Voices change throughout life. Children have higher-pitched voices due to shorter vocal cords. During puberty, hormones, particularly testosterone in males, cause the larynx to grow and the vocal cords to lengthen and thicken, lowering pitch. In older age, the vocal cords can become stiffer and weaker, leading to changes in voice quality. The ears that receive speech are marvels of mechanical and neural engineering. Sound waves funnel into the ear canal and strike the tympanic membrane, the eardrum. This thin membrane vibrates in response to sound, and these vibrations are transmitted through the three tiny bones of the middle ear. These bones, the ossicles, form a chain that amplifies the vibrations and delivers them to the cochlea in the inner ear. The cochlea is a fluid-filled spiral structure about the size of a pea. Inside it runs the basilar membrane, along which sit rows of hair cells, the sensory receptors for hearing. These hair cells have tiny hair-like projections called stereocilia that extend into the fluid. When vibrations from the ossicles create waves in the cochlear fluid, the basilar membrane moves, causing the stereocilia to bend. This bending opens ion channels, generating electrical signals that travel along the auditory nerve to the brain. Different frequencies of sound cause different parts of the basilar membrane to vibrate most strongly. High-frequency sounds affect the base of the cochlea, near the entrance, while low-frequency sounds affect the apex, deeper in the spiral. This spatial mapping of frequency is preserved all the way to the auditory cortex, where neurons are organized tonotopically, responding preferentially to specific frequencies. The auditory system is extraordinarily sensitive. The quietest sound a healthy human ear can detect causes the eardrum to move a distance smaller than the diameter of a hydrogen atom. At such sensitivity, the ear is nearly at the limit imposed by random thermal motion of air molecules. If hearing were any more sensitive, the constant background noise of molecular collisions would be audible. Balance, too, is mediated by the inner ear, though by different structures. Adjacent to the cochlea are three semicircular canals, oriented at right angles to each other, corresponding to the three dimensions of space. These canals are filled with fluid, and at one end of each canal is a swelling containing hair cells embedded in a gelatinous structure. When the head rotates, the fluid in the corresponding canal lags behind due to inertia. bending the hair cells and signaling the direction and speed of rotation. Two other structures, the utricle and saccule, detect linear acceleration and the orientation of the head relative to gravity. They contain hair cells with tiny calcium carbonate crystals, called otoliths, resting on them. When the head tilts or accelerates, gravity or inertia causes the otoliths to shift, bending the hair cells. Together, the semicircular canals, utricle, and saccule provide comprehensive information about head position and motion, which the brain integrates with visual and proprioceptive information to maintain balance. Proprioception, the sense of body position, comes from receptors in muscles, tendons, and joints that detect stretch, tension, and pressure. These receptors constantly inform the brain about the position of limbs and the state of muscle contraction. This allows movement to be coordinated without constant visual monitoring. You can touch your nose with your eyes closed, walk in the dark without falling, type without looking at your fingers. Proprioception creates an internal map of the body, a sense of where you are in space. This sense can be disrupted by neurological conditions or injury. People with proprioceptive loss describe feeling disconnected from their bodies, having to watch their limbs to know where they are, struggling with tasks that require fine motor control. It reveals how much of normal movement relies on unconscious sensory feedback, constant adjustments happening beneath awareness. Touch, too, is more complex than it might seem. The skin contains multiple types of receptors, each specialized for different aspects of tactile sensation. Merkel cells detect sustained pressure and fine details, allowing you to read braille or feel textures. Meissner's corpuscles respond to light touch and low-frequency vibration, sensitive to changes in texture. Pacinian corpuscles detect deep pressure and high-frequency vibration, like the feeling of holding a vibrating phone. Ruffini endings respond to skin stretch, providing information about finger position and movement. Pain receptors, or nociceptors, are different. They are free nerve endings that respond to potentially damaging stimuli. Extreme heat or cold, intense pressure, chemical irritants. Pain signals travel along two types of nerve fibers. Fast fibers transmit sharp, localized pain quickly, triggering immediate reflexive withdrawal. Slow fibers carry dull, aching pain that lingers, serving as a reminder to protect the injured area. Pain is not just a sensory experience, but also an emotional one, involving brain regions associated with distress and aversion. Chronic pain can alter these circuits, leading to persistent suffering even after the original injury has healed. Temperature sensation also involves specialized receptors. Some respond to warmth, others to cold. Extreme temperatures activate the same nociceptors that signal pain, which is why very hot and very cold can both be painful. The sensation of temperature is relative. If you place one hand in cold water and the other in hot water, then place both in lukewarm water, the lukewarm water will feel hot to the cold hand and cold to the hot hand. The brain interprets temperature based on contrast and recent experience, not absolute values. Smell and taste, the chemical senses, are closely related. Olfactory receptors in the nasal cavity detect airborne molecules. When you smell something, volatile molecules from that substance enter the nose and bind to receptors on olfactory neurons. These neurons send signals directly to the olfactory bulb, part of the brain, bypassing the thalamus, which is unusual. From the olfactory bulb, signals are sent to several brain regions, including the amygdala and hippocampus, which are involved in emotion and memory. This direct connection is why smells can trigger vivid memories and strong emotional responses. Humans have about 400 types of olfactory receptors, far fewer than dogs, but still sufficient to distinguish an enormous variety of scents. Each receptor responds to certain molecular features, and the combination of receptors activated by a given odor creates a unique pattern that the brain interprets as a specific smell. Recent estimates suggest humans can distinguish over 1 trillion different odors, far more than previously thought. Taste is mediated by taste buds, clusters of sensory cells on the tongue, and other parts of the mouth. Traditionally, 5 basic tastes are recognized: sweet, salty, sour, bitter, and umami, a savory taste associated with amino acids like glutamate. Each taste signals something about the nutritional or safety content of food. Sweet indicates energy-rich carbohydrates. Salty signals essential electrolytes. Sour often indicates acidity, which can signal spoilage. Bitter warns of potential toxins. Many poisonous plants taste bitter. Umami indicates protein. What we perceive as flavor is actually a combination of taste and smell, along with texture, temperature, and even visual appearance. This is why food tastes bland when you have a stuffy nose. The olfactory component is missing. Much of the richness of flavor comes from aromas released while chewing that travel up the back of the throat to the nasal cavity, stimulating olfactory receptors. This retronasal olfaction is distinct from smelling through the nostrils, but uses the same receptors. The brain integrates all these sensory modalities into a unified perceptual experience. This integration is not seamless or instantaneous. Signals from different senses travel at different speeds and are processed in different brain regions. The brain must synchronize them, compensating for differences in transmission time. Visual information takes longer to process than auditory information, yet we perceive sights and sounds as occurring simultaneously. The brain creates this illusion of synchrony by holding faster signals briefly, waiting for slower ones. This integration can be fooled. The ventriloquism effect occurs when vision and sound are spatially misaligned. The brain often resolves this by perceiving the sound as coming from the visual source, which is why you perceive the ventriloquist's dummy as speaking even though the sound comes from the ventriloquist. The McGurk effect demonstrates cross-modal interaction in speech perception. If you hear one syllable while watching a video of someone mouthing a different syllable, you may perceive a third syllable, a fusion of the auditory and visual information. These illusions reveal that perception is not a passive recording but an active construction. The brain predicts what it expects to perceive based on context and past experience, then adjusts based on incoming sensory data. Most of the time, these predictions are accurate, allowing rapid and efficient perception. When predictions fail, illusions, hallucinations, or perceptual errors can result. Hallucinations can occur in the absence of sensory input or through misinterpretation of ambiguous stimuli. They are common in certain medical conditions, such as schizophrenia, Parkinson's disease, or Charles Bonnet syndrome, where vision loss leads to visual hallucinations, as the brain generates imagery to fill the sensory void. Hallucinations can also be induced by drugs, fever, sleep deprivation, or sensory isolation. The brain is constantly generating activity, and without external input to constrain it, internal activity can be mistaken for external reality. Dreams are a form of internally generated experience, occurring during sleep when external sensory input is minimal. During REM sleep, the brain is highly active, but the thalamus blocks most sensory signals from reaching the cortex, and motor signals to the muscles are inhibited, causing temporary paralysis. This prevents the dreamer from acting out the dream, though the brain still generates motor commands. The content of dreams draws on memories, concerns, recent experiences, but assembled in ways that often defy logic or physical possibility, hybrid state between ordinary dreaming and waking consciousness. Some people can learn to induce lucid dreams through practice, using reality checks and other techniques to recognize when they are dreaming. Sleep itself is not a uniform state, but a cycle of stages. Non-REM sleep is divided into 3 stages, from light sleep in stage 1 to deep slow-wave sleep in stage 3. During slow-wave sleep, the brain's electrical activity shows large, synchronized oscillations, and it is during this stage that growth hormone is released, supporting tissue repair and growth. REM sleep, characterized by rapid eye movements and vivid dreams, occurs cyclically, approximately every 90 minutes, with REM periods becoming longer toward morning. The purpose of sleep extends beyond rest. During sleep, the glymphatic system, a waste clearance system in the brain, becomes more active. Cerebrospinal fluid flows through the brain, flushing out metabolic waste products that accumulated during waking hours, including beta amyloid, a protein associated with Alzheimer's disease. This cleansing process is thought to be one reason why chronic sleep deprivation is associated with cognitive decline and increased risk of neurodegenerative diseases. Sleep deprivation has widespread effects on the body and mind. Cognitive functions decline, attention wavers, reaction times slow, memory consolidation is impaired. Mood is affected, with increased irritability, anxiety, and susceptibility to stress. The immune system weakens, making infections more likely. Metabolic regulation is disrupted, increasing hunger and impairing glucose metabolism, which over time can contribute to weight gain and diabetes. Even a single night of poor sleep has measurable effects, though most are reversed with recovery sleep. Chronic sleep deprivation is alarmingly common in modern society, driven by artificial lighting, work schedules, electronic devices, and social demands. Blue light emitted by screens suppresses melatonin production, delaying sleep onset. The human circadian system evolved over millions of years in environments where light and darkness followed the sun. Modern lifestyles disrupt this ancient rhythm, with consequences still being understood. The circadian system coordinates not just sleep, but a wide array of physiological processes. Body temperature follows a circadian rhythm, lowest in the early morning, highest in late afternoon. Hormone levels fluctuate. Cortisol peaks in the morning to promote wakefulness. Melatonin rises in the evening to promote sleep. Immune function, cell division, even gene expression vary with time of day. Nearly every cell in the body contains molecular clock machinery, genes that oscillate with a roughly 24-hour period, turned on and off in rhythmic cycles. These cellular clocks are synchronized by the master clock in the suprachiasmatic nucleus, which itself is set by light exposure. When light hits the retina, a subset of retinal ganglion cells that contain melanopsin, a light-sensitive protein, sends signals to the suprachiasmatic nucleus, informing it of the light-dark cycle. This allows the internal clock to align with the external day. In the absence of light cues, the circadian rhythm free runs with a period slightly longer than 24 hours, which is why blind individuals or people in constant darkness gradually shift their sleep-wake cycles. Jet lag occurs when travel rapidly shifts the external time zone, but the internal circadian system lags behind, taking several days to adjust. The symptoms: fatigue, insomnia, digestive issues, result from the mismatch between the internal clock and the external environment. Shift workers face chronic circadian disruption, working when their bodies expect to be awake. This chronic misalignment is associated with increased risk of cardiovascular disease, metabolic disorders, mood disorders, and certain cancers. Cancer arises when cells lose normal growth regulation and divide uncontrollably. Every cell division carries a small risk of error, mutations in the DNA that can alter gene function. Most mutations are harmless or corrected by cellular repair mechanisms. Some cause the cell to die, but occasionally, a mutation occurs in a gene that regulates cell growth or suppresses tumor formation. If additional mutations accumulate over time, disabling multiple safeguards, a cell may become cancerous. Cancer is not a single disease, but a collection of diseases characterized by uncontrolled cell growth. Different cancers arise from different cell types and have different behaviors. Some grow slowly, remaining localized for years. Others grow rapidly and metastasize, spreading to distant parts of the body via the bloodstream or lymphatic system. Once metastasized, cancer becomes far more difficult to treat, as cancer cells establish new tumors in organs far from the original site. The immune system normally detects and destroys abnormal cells, including early cancer cells. Immune cells patrol the body, identifying cells that display abnormal proteins on their surface and eliminating them. But cancer cells can evolve mechanisms to evade immune detection, disguising themselves as normal or actively suppressing immune responses. Cancer immunotherapy, a relatively new treatment approach, aims to enhance the immune system's ability to recognize and attack cancer cells. Traditional cancer treatments include surgery, radiation, and chemotherapy. Surgery removes tumors when they are accessible and localized. Radiation uses high-energy particles or waves to damage the DNA of cancer cells, killing them or preventing them from dividing. Chemotherapy uses drugs that target rapidly dividing cells, which includes cancer cells but also affects normal cells that divide rapidly, such as those in the bone marrow, digestive tract, and hair follicles, leading to side effects like anemia, nausea, and hair loss. The body's capacity for healing and regeneration varies widely by tissue. Some tissues, like the liver and skin, regenerate readily. Others, like cardiac muscle and neurons, have limited regenerative capacity. When a heart attack damages cardiac muscle, the dead tissue is replaced not with new muscle cells, but with scar tissue, which cannot contract. This reduces the heart's pumping efficiency. Similarly, when neurons in the brain or spinal cord die, they are not replaced, which is why strokes and spinal cord injuries often result in permanent deficits. Yet, even in these tissues, there is some capacity for recovery through plasticity and compensatory mechanisms. After a stroke damages part of the brain, Other regions can sometimes take over lost functions, especially with rehabilitation. The extent of recovery depends on the location and size of the injury, the age of the patient, and the intensity of rehabilitation. Younger brains are generally more plastic, but even older adults can show remarkable recovery. Aging itself is a gradual accumulation of damage and loss of function. Cells become less efficient at repairing DNA. Telomeres, protective caps on the ends of chromosomes, shorten with each cell division, eventually limiting further division. Proteins accumulate damage from oxidative stress, misfolding and clumping. Mitochondria become less efficient, producing less ATP and more harmful reactive oxygen species. Stem cells become less active, reducing the body's regenerative capacity. These changes manifest in various ways. Skin loses elasticity as collagen and elastin fibers degrade. Hair grays as melanocytes in hair follicles stop producing pigment. Bones lose density, becoming more fragile. Muscles shrink, losing strength and mass. Joints stiffen as cartilage wears down. Vision declines as the lens loses flexibility, making it harder to focus on close objects. A condition called presbyopia. Hearing diminishes, particularly for high frequencies, as hair cells in the cochlea are damaged and not replaced. Cognitive changes also occur, though they vary widely between individuals. Processing speed tends to slow. Short-term memory may become less reliable, but accumulated knowledge and vocabulary often remain stable or even improve. Wisdom, if it can be defined, may increase with age, as decades of experience provide context and perspective. Not all cognitive decline is inevitable. Physical exercise, mental stimulation, social engagement, and a healthy diet can all support cognitive function in later life. The maximum human lifespan appears to be around 120 years, though very few individuals reach this age. Most people die far earlier from disease, injury, or the accumulated effects of aging. Throughout most of human history, life expectancy was low, often less than 40 years, due to high infant mortality, infectious disease, and lack of medical care. In the past century, life expectancy has increased dramatically in many parts of the world due to improvements in sanitation, nutrition, antibiotics, vaccines, and medical technology. Yet death remains inevitable. Cells wear out. Systems fail. The body's complex regulatory mechanisms, maintained through constant expenditure of energy, eventually break down. Entropy, the tendency toward disorder, is a fundamental law of thermodynamics, and living things are no exception. Life is a temporary state of highly organized complexity, maintained only through continuous input of energy and constant repair. When these processes can no longer keep pace with damage, the system collapses. What happens at the moment of death depends on the cause. If the heart stops, blood flow ceases, and within seconds, the brain is deprived of oxygen. Consciousness fades rapidly. Brain cells begin to die within minutes. If breathing stops, the result is similar: hypoxia leading to loss of consciousness, and then brain death. Cells throughout the body continue to live for varying amounts of time after death. Skin cells can survive for hours. Some tissues can be harvested for transplantation even after cardiac death. Decomposition begins shortly after death, as the body's own enzymes and bacteria start breaking down tissues. In life, the immune system and other defenses keep these processes in check. After death, these constraints are removed. The microbiome, which lived symbiotically during life, now consumes the body that hosted it. Over time, soft tissues break down, leaving only bones, which can persist for centuries or millennia in the right conditions, eventually becoming fossils if mineralized. Fossils provide a record of life's history on Earth, a history stretching back more than 3.5 billion years. The earliest fossils are stromatolites, layered structures created by photosynthetic bacteria. For most of Earth's history, life consisted only of single-celled organisms, bacteria and archaea, complex cells with nuclei, eukaryotes, appeared around 2 billion years ago, possibly arising from symbiotic relationships between different types of bacteria. Multicellular life is a more recent innovation, appearing around 600 million years ago. The Cambrian explosion, a rapid diversification of life forms beginning around 540 million years ago, saw the emergence of most major animal body plans. Vertebrates, animals with backbones, arose during this time, initially as small fish-like creatures. Over hundreds of millions of years, vertebrates diversified into fish, amphibians, reptiles, birds, and mammals. Mammals evolved from reptilian ancestors around 200 million years ago, during the age of dinosaurs. Early mammals were small, nocturnal creatures, likely eating insects and staying out of the way of the dominant reptiles. When a massive asteroid struck Earth 66 million years ago, triggering mass extinctions that wiped out the non-avian dinosaurs, mammals survived and rapidly diversified to fill vacant ecological niches. Primates, the group that includes lemurs, monkeys, apes, and humans, emerged around 60 million years ago. The earliest primates were small, tree-dwelling animals with grasping hands and forward-facing eyes, adaptations for life in the forest canopy. Over time, primates diversified. Some lineages became larger and more terrestrial. The great apes, which include orangutans, gorillas, chimpanzees, and humans, share a common ancestor that lived around 15 million years ago. The human lineage split from the chimpanzee lineage around 6 to 7 million years ago in Africa. The earliest hominins, human ancestors, were still ape-like but showed adaptations for bipedal walking, moving on 2 legs rather than 4. This freed the hands for carrying objects and tools, and it changed the shape of the skull, pelvis, and spine. Why bipedalism evolved is still debated, but it was a defining characteristic of hominins. Over millions of years, hominin brains gradually increased in size. Early species like Australopithecus had brains similar in size to chimpanzees. Later species, including Homo habilis and Homo erectus, had larger brains and began making and using stone tools. Homo erectus was the first hominin to leave Africa, spreading into Asia and Europe over 1 million years ago. Anatomically modern humans, Homo sapiens, emerged in Africa around 300,000 years ago. For most of this time, humans lived as hunter-gatherers in small mobile groups, foraging for plants and hunting animals. The development of language, symbolic thought, and culture allowed humans to cooperate in large groups, share knowledge, and adapt to diverse environments. Around 70,000 years ago, humans began migrating out of Africa, eventually colonizing every continent except Antarctica. This global dispersal required adaptation to vastly different climates and ecosystems, from tropical rainforests to Arctic tundra. Humans developed technologies like clothing, fire, shelters, and complex tools to survive in these varied environments. The invention of agriculture around 10,000 years ago marked a major turning point. Instead of moving to find food, humans began cultivating crops and domesticating animals, allowing them to settle in permanent villages. Agriculture supported larger populations and led to the development of complex societies, specialization of labor, writing, cities, and states. It also brought new challenges. Denser populations facilitated the spread of infectious diseases. Social hierarchies led to inequality, and dependence on crops made communities vulnerable to drought and famine. Infectious diseases have shaped human history profoundly. Smallpox, plague, typhus, influenza, and countless other pathogens have caused devastating epidemics. The Black Death in the 14th century killed an estimated 1/3 of Europe's population. The 1918 influenza pandemic killed tens of millions worldwide. These diseases exploit the body's vulnerabilities, evolving rapidly to evade immune defenses and spread efficiently between hosts. Pathogens include viruses, bacteria, fungi, and parasites. Viruses are not technically alive. They are packages of genetic material, DNA or RNA, wrapped in a protein coat. They cannot reproduce on their own, but must hijack a host cell's machinery. A virus attaches to a cell, injects its genetic material, and uses the cell's ribosomes and enzymes to produce new viral particles, which then burst out, killing the cell, and infect others. Bacteria are single-celled organisms, much larger and more complex than viruses. Some bacteria are beneficial, essential components of the microbiome, but others cause disease. Pathogenic bacteria produce toxins, damage tissues, and trigger inflammatory responses. Before antibiotics, bacterial infections were a leading cause of death. The discovery of penicillin in the early 20th century revolutionized medicine, but bacteria have evolved resistance to many antibiotics, posing a growing threat. The immune system defends against pathogens through multiple layers. Physical barriers like skin and mucous membranes prevent entry. If pathogens breach these barriers, the innate immune system responds rapidly. White blood cells called neutrophils and macrophages engulf and destroy invaders. Natural killer cells target virus-infected cells. Complement proteins mark pathogens for destruction. If the innate response is insufficient, the adaptive immune system activates. This takes longer but is highly specific. B cells produce antibodies, proteins that bind to specific antigens on the pathogen's surface, neutralizing it or marking it for destruction. T cells come in several types. Helper T cells coordinate the immune response, killer T cells destroy infected cells, and regulatory T cells prevent overreaction. After the infection is cleared, memory B and T cells remain, providing long-lasting immunity. Vaccines work by exposing the immune system to a harmless version of a pathogen, dead, weakened, or just a fragment, allowing it to develop memory without causing disease. When the real pathogen is encountered, the immune system responds swiftly and effectively. Vaccination has eradicated smallpox and nearly eliminated polio, saving countless lives. But the immune system can malfunction. Autoimmune diseases occur when it attacks the body's own tissues, mistaking them for foreign. In type 1 diabetes, immune cells destroy insulin-producing cells in the pancreas. In multiple sclerosis, they attack the myelin sheath around neurons. In rheumatoid arthritis, they target joints. The causes of autoimmune diseases are not fully understood, but likely involve a combination of genetic susceptibility and environmental triggers. Allergies are another immune malfunction. Hypersensitive reactions to harmless substances like pollen, certain foods, or pet dander. The immune system treats these as threats, producing antibodies that trigger release of histamine and other chemicals, causing symptoms like sneezing, itching, swelling, or in severe cases, anaphylaxis, a life-threatening reaction. The immune system's complexity reflects the constant evolutionary arms race between hosts and pathogens. Pathogens evolve to evade immune defenses, and immune systems evolve counter-strategies. This dynamic has been ongoing for hundreds of millions of years, shaping both hosts and pathogens in profound ways. Evolution operates through variation and selection. Random mutations introduce genetic variation. Individuals with traits that enhance survival and reproduction in a given environment are more likely to pass those traits to the next generation. Over time, beneficial traits become more common and populations adapt. This process, natural selection, is the engine of evolution. Natural selection does not have foresight or purpose. It is simply the differential survival and reproduction of individuals based on heritable traits. It cannot create perfection, only incremental improvement relative to the current environment. Many features of organisms are not optimal but good enough, constrained by evolutionary history, developmental processes, and trade-offs. The human body bears many marks of its evolutionary past. The appendix, a small pouch attached to the large intestine, may have once played a role in digesting plant material, but now serves little function and can become inflamed, causing appendicitis. Wisdom teeth, the third molars, were useful when human diets included coarser food, requiring more chewing, but modern jaws are often too small to accommodate them, leading to impaction and the need for removal. The recurrent laryngeal nerve, which controls muscles in the larynx, takes an absurdly long path, looping down around the aorta before returning to the larynx. A route that makes sense only in light of evolutionary history, inherited from fish ancestors where the heart was positioned differently. These quirks are evidence of evolutionary tinkering, building on what already exists rather than designing from scratch. Evolution has no designer, no blueprint, only the blind process of variation and selection, playing out over immense timescales. Yet from this process has emerged extraordinary complexity, eyes that detect single photons, brains that ponder their own existence, hands that create art and tools, hearts that beat billions of times without rest. The body is not a machine, though it is often described as one. Machines are designed, built from separate parts according to a plan. The body is grown, emerging from a single cell through processes of self-organization and differentiation. It is not static but constantly changing, not perfectly efficient but remarkably resilient, not assembled but generated, layer upon layer, system upon system, woven together in ways that are still not fully understood. Understanding the body has been one of humanity's great endeavors, spanning millennia. Ancient civilizations developed medical knowledge through observation and trial and error. The Egyptians performed surgeries and mummification, providing early anatomical knowledge. The Greeks, particularly Hippocrates and Galen, attempted to systematize medicine, though their theories were often incorrect. Galen's ideas dominated for over 1,000 years until the Renaissance, when anatomists like Vesalius began dissecting human bodies and correcting ancient errors. The discovery of the circulatory system by William Harvey in the 17th century was revolutionary, demonstrating that blood circulates in a closed loop pumped by the heart, contradicting the long-held belief that blood was consumed by tissues and continuously produced. The invention of the microscope opened up the world of cells and microorganisms, invisible to the naked eye but fundamental to life and disease. The germ theory of disease, developed in the 19th century by scientists like Pasteur and Koch, established that many diseases are caused by microorganisms, not by imbalanced humors or miasmas. This led to practices like sterilization, antiseptics, and eventually antibiotics, transforming medicine and public health. The 20th century brought rapid advances: the discovery of DNA's structure, the development of vaccines and antibiotics, imaging technologies like X-rays, CT scans, and MRI that allow non-invasive views inside the body, organ transplantation, and the beginning of genetic medicine. The Human Genome Project, completed in 2003, sequenced all 3 billion base pairs of human DNA, providing a reference map of the human genetic code. Yet despite all this knowledge, much remains mysterious. How exactly does consciousness arise from neural activity? What determines why some people develop diseases while others with similar genetics and environments do not? How can aging be slowed or reversed? Can lost neurons be regenerated? These questions drive ongoing research, pushing the boundaries of what is known. The body continues to reveal itself, slowly, in layers. Each answer opens new questions. Each technique unveils new complexities. The more closely it is examined, the more intricate it becomes. Organs are made of tissues of cells, cells of organelles, organelles of molecules of atoms. At each level, new properties emerge that cannot be fully predicted from the level below. Life is not just chemistry, though it is chemical. It is organization, pattern, process, dynamic stability maintained far from equilibrium. Equilibrium, in thermodynamic terms, is death. A living organism maintains internal conditions far from equilibrium with its environment, expending energy to do so. It concentrates certain molecules, maintains pH within narrow ranges, regulates temperature, pumps ions against gradients. All of this requires energy, ultimately derived from the sun through the chain of photosynthesis, food consumption, digestion, and cellular respiration. Energy flows through ecosystems, from sunlight captured by plants, to herbivores that eat plants, to carnivores that eat herbivores, to decomposers that break down dead matter. At each step, some energy is lost as heat. The second law of thermodynamics ensures that energy transformations are never perfectly efficient. Entropy always increases, but life locally decreases entropy by creating order. paying for it with increased entropy elsewhere. In the heat radiated, the waste excreted, the resources consumed. Humans have become incredibly efficient at extracting energy from the environment, far beyond what any other species can match. Agriculture, fossil fuels, technology, all amplify the energy available per person. This has allowed human populations to grow exponentially, from a few million in prehistory to billions today. But this growth comes with consequences. Resource depletion, habitat destruction, pollution, climate change. The human body evolved in environments vastly different from modern industrial societies. For most of human evolution, food was scarce and required effort to obtain. The body adapted to store energy as fat during times of plenty, preparing for times of scarcity that no longer come. Physical activity was necessary for survival, hunting, gathering, escaping predators. Now, in much of the world, food is abundant and physical activity optional, leading to epidemics of obesity, diabetes, cardiovascular disease. Modern environments present challenges the body was not evolved to handle. Chronic stress from work, social pressures, constant connectivity activates the fight or flight response, designed for short-term physical threats, flooding the body with stress hormones like cortisol and adrenaline. When sustained over long periods, this contributes to hypertension, weakened immune function, anxiety, and depression. Artificial light disrupts circadian rhythms. Processed foods high in sugar, salt, and unhealthy fats hijack reward systems evolved to seek out rare, calorie-dense foods. Sedentary lifestyles lead to muscle atrophy and bone loss. Social isolation, despite being more connected than ever through technology, can impair mental and physical health. As humans are deeply social creatures whose brains and bodies evolved in close-knit communities, yet the body is adaptable. Physical exercise stimulates muscle growth, strengthens bones, improves cardiovascular health, enhances mood. Adequate sleep allows the brain to rest and consolidate memories. Nutritious food provides the raw materials for cellular repair. Social connection, meaningful work, time in nature, all support well-being in ways that align with our evolutionary heritage. The body is not infinitely resilient, but it is remarkably forgiving. It can recover from injury, adapt to stress, compensate for loss. It is a system with redundancies, backups, feedback loops that maintain stability. When one kidney fails, the other compensates. When lung capacity is reduced, breathing rate increases. When neurons die, others can sometimes take over their functions. The body seeks balance, homeostasis, constantly adjusting to internal and external changes. This balancing act is ongoing, moment to moment, never complete, never finished. Blood sugar rises after a meal. Insulin is released. Cells take up glucose. Blood sugar falls. Glucagon is released. Glucose is released from storage. Body temperature fluctuates. Cooling mechanisms activate or shut down. Carbon dioxide levels shift. Breathing adjusts. Thousands of such feedback loops operate simultaneously, interacting, influencing each other, creating a dynamic stability that from the outside appears as the simple fact of being alive. but not unchanging. The body this moment is not the same as the body a moment ago. Cells have divided, molecules have been synthesized and broken down, electrical signals have fired, blood has circulated. Over longer timescales, the changes accumulate. The infant becomes the child becomes the adult ages. Features change, abilities wax and wane, the body rewrites itself continuously. And within this continuous rewriting, there is something that persists, a sense of self, of identity continuous through time, despite the physical changes. The molecules that composed the body years ago have mostly been replaced, yet memory remains. Personality persists. The thread of subjective experience continues. What constitutes the self is not the material substance, but the pattern, the organization, the process. The self is not a thing but a happening, a verb more than a noun, a flow of information and experience through a constantly renewing physical substrate. This flow does not occur in isolation. The body is embedded in an environment, shaping it in turn. Breath links the internal and external, air flowing in and out, oxygen and carbon dioxide exchanged, connecting lungs to atmosphere. Food and water cross the boundary, becoming part of the body, their atoms rearranged into cells and tissues. Heat radiates outward, sounds emerge from the vocal cords, vibrations in air, carrying meaning. Light reflects off the skin, creating visual appearance. The boundary between self and world is permeable, ambiguous. Even the microbiome blurs this boundary. Are the bacteria in the gut part of the self? They are not human cells, yet they are essential for health, integrated into bodily functions. They have their own genetic material, their own evolutionary histories, yet they cannot survive without the host, and the host cannot thrive without them. The body is not a single organism but an ecology, a community of living entities in dynamic relationship. This ecological view extends outward. Humans exist within ecosystems, dependent on plants for food and oxygen, on microbes for nutrient cycling, on insects for pollination, on predators and prey in complex food webs. The health of the body is inseparable from the health of the environment. Contaminated water, polluted air, degraded soil, all impact human health directly. The body breathes the air, drinks the water, consumes the products of the land, and the land itself is alive in a sense, not as a single organism but as a system of flows and cycles. Water evaporates from oceans, falls as rain, flows through rivers, seeps into groundwater, returns to the sea. Carbon cycles between atmosphere, living organisms, and geological formations. Nitrogen is fixed by bacteria, taken up by plants, consumed by animals, returned to soil. These cycles have operated for billions of years, creating and maintaining conditions conducive to life. Life, in turn, has shaped the planet. Photosynthetic organisms produced the oxygen-rich atmosphere that allowed complex life to evolve. Plants and microbes weather rocks, creating soil. Coral reefs build massive structures. Human activity now alters the planet on geological scales, changing atmospheric composition, transforming landscapes, driving species to extinction. The body, this small collection of organized matter, is part of these vast planetary processes. A temporary local decrease in entropy, sustained by flows of energy and matter through systems within systems, from molecules to cells to organs to organisms to ecosystems to the biosphere. At every scale, there are patterns, structures emerging from the interactions of simpler components, behaving in ways not predictable from those components alone. This emergence is one of the deepest mysteries. How do mindless molecules arranged in certain ways give rise to life, to sensation, to thought, to the feeling of being? The question remains open, perhaps ultimately unanswerable in any complete sense. Yet the exploration continues, layer upon layer, deeper and broader, seeking understanding of this strange phenomenon of being a body, of being in a body, of being at all. And in this seeking, in this wondering, the body sits, stands, lies down, breathes, pulses, senses, moves through space and time, carrying within it a history stretching back to the first stirrings of life in ancient seas, forward to futures unknown, existing. It just lives. It breathes, repairs, remembers. It holds you through stillness and change. It asks for rest and keeps watch as you drift. Out there, the world turns. Inside, your own galaxies shift. Cells dividing, currents flowing, signals pulsing in rhythms too small to name. Even in sleep, you are vast. So let your shoulders soften. Let your breath deepen. There's nothing you need to do now. The body will carry you the rest of the way. And so, under the quiet stars, the story continues. Hope you sleep well and dream softly. Good night.